Australia has spent 90 years losing a war it never meant to start. The enemy weighs less than a bag of sugar, secretes poison strong enough to stop a dog's heart, and has bulldozed its way across an entire continent almost unopposed. Snakes died trying to eat it. Goannas died trying to eat it. Quolls, freshwater crocodiles, even family pets. All of them learned the same lesson the hard way. And then on the muddy edge of a tropical waterhole, scientists watched something no textbook had prepared them for. An insect roughly the size of a grain of rice, walking straight up to that invincible poison factory and taking it apart alive. The insect is a meat ant. The victim is a baby cane toad. And the fight between them may be the most promising native answer Australia has ever found to one of the worst biological invasions in its history. To understand why this discovery matters, you first have to understand just how untouchable cane toads have been. Cane toads, scientifically Rhinella marina, long classified as Bufo marinus, carry bufadienolide toxins in glands across their backs and shoulders. These compounds interfere directly with the cardiac and nervous systems of most vertebrates. A native predator that has never encountered the toad before, a snake, a lizard, a marsupial, typically has no defense mechanism against it. One bite is often fatal. That vulnerability comes from a simple evolutionary mismatch. Cane toads evolved in Central and South America, alongside predators that, over millions of years, developed some tolerance to their toxins. Australia's predators never had that chance. When the toads arrived, native wildlife treated them like any other slow, defenseless amphibian. An easy meal. It wasn't. Research groups tracking the invasion found that in some regions, populations of native predators, such as northern quolls and large monitor lizards, collapsed by more than 90% within a few years of cane toads moving through. But researchers at the University of Sydney, working under a research group informally known as Team Bufo, and led by ecologist Professor Rick Shine, noticed something different happening at the water's edge. A large, aggressive native ant, the meat ant Iridomyrmex reburus, was regularly seen killing and eating baby cane toads, seemingly without any ill effects. Meat ants are common across tropical northern Australia, where they build large surface nest colonies and forage aggressively during the day. Unlike vertebrates, most invertebrates have little to no sensitivity to the bufadienolide toxins that make cane toads so dangerous to snakes, birds, and mammals. That single biological fact, immunity by accident of biology, not adaptation, is the entire reason this story is possible. In 2009, honors student Georgia Wardfear, working with Professor Shine, Gregory Brown, and Matthew Greenlees, published research in the journal Functional Ecology comparing the behavior of meat ants, baby cane toads, and 7 species of native Australian frog. What they found was a textbook case of what ecologists call a maladaptive trait. Native frog species were nocturnal, sheltering in vegetation during the day, precisely when meat ants were most active and hunting. Cane toad metamorphs, the tiny newly transformed toadlets emerging from the water, did the opposite. They were active by day, and they preferred open, exposed ground at the very edges of ponds, which happens to be exactly where meat ant colonies forage most intensively. Worse for the toads, their entire defensive strategy backfired. When threatened, a cane toad's instinct, inherited from a home range where predators respect the poison, is to freeze in place and let its toxins do the work. It is a strategy that has protected the species for millions of years in South America. It does nothing against an attacker with no capacity to be poisoned. As the research team put it, native frogs had learned or evolved avoidance behavior around meat ants. Cane toads simply had not. The toads sat still while ants clamped onto their legs, recruited reinforcements, and dismantled them on the spot. To appreciate why an ant colony finally moving the needle is such a big deal, it helps to rewind to where this all started. In 1935, sugarcane growers in Queensland were fighting a real and costly problem. Native cane beetle larvae, including the grayback and Frenchy beetle species, were chewing through the root systems of sugarcane crops. The Queensland Bureau of Sugar Experimentations sent entomologist Reginald Montgomery to Hawaii, where a South American toad species had reportedly helped control beetle pests in sugarcane fields there. In June 1935, Montgomery collected 102 toads, 51 males and 51 females, packed them into wood shaving line crates, and shipped them back to an experimental station at Moringa, near Gordonvale in far north Queensland. The toads bred rapidly in captivity. By August 1935, roughly 2,400 juvenile toads were released into cane fields around Gordonvale. No environmental impact studies were conducted beforehand, and nobody had confirmed the toads would even eat the target beetles. They didn't. The grayback beetle spends most of its life high on the cane stalks, well out of reach of a ground-dwelling toad. The two species barely cross paths. What the toads did do was eat almost everything else within striking distance: insects, native frogs, small reptiles, and carrion, and breed at an extraordinary rate. A single female cane toad can lay between 8,000 and 30,000 eggs in one clutch, sometimes more than once a year. From that founding population of roughly 100 animals, Australia's cane toad population is now estimated at well over 200 million, spread across Queensland, the Northern Territory, Western Australia, and into New South Wales. The invasion front itself has been accelerating. In the early decades after release, toads expanded their range by roughly 10 kilometers a year. Research published in Nature in 2006 by Benjamin Phillips, Gregory Brown, Jonathan Webb, and Rick Shine found that toads at the leading edge of the invasion had evolved measurably longer legs than toads in long-established populations. Legs making up about 45% of body length at the front, compared with roughly 35% further back. Longer-legged toads could travel farther and faster, and because the fastest toads at the front were the ones breeding with each other, that trait compounded across generations. By the mid-2000s, the invasion front was advancing roughly 5 times faster than it had 60 years earlier, up to 50 kilometers a year in some areas. Every conventional tool thrown at the problem struggled to keep pace. Government-built toad exclusion fencing, community toad-busting nights, where volunteers collected and humanely euthanized toads by the thousands, acoustic traps designed to mimic toad mating calls, and experimental efforts to condition native predators to avoid toads through controlled, non-lethal toxin exposure. All of these produced localized benefits, but none scaled to match a population spreading across a continent. The cane toad had, for decades, been treated as an unstoppable, structural feature of the Australian landscape. That is what makes the meat ant findings so significant. This wasn't an imported solution, a genetically engineered animal, or an expensive infrastructure project. It was a native species that had been living alongside toad-invaded waterholes the entire time, already doing part of the job, just not in high enough numbers, in exactly the right place, at exactly the right moment. Knowing that meat ants killed baby toads was one thing. Turning that into an actual control method was another. The problem researchers faced was straightforward. Meat ant density varies a lot from pond to pond, and toads were often emerging in numbers or in locations where ant colonies simply weren't concentrated enough to make a dent. Ward Fear and colleagues tested a strikingly low-cost solution: bait. In a 2010 study published in the Journal of Applied Ecology, the team placed open cans of ordinary fish-based cat food along the margins of ponds, where cane toad metamorphs were expected to emerge. The strong smell drew meat ants out of their colonies in large numbers, holding them at the water's edge for extended periods rather than their usual pattern of foraging in bursts and retreating. The results were dramatic. Baiting with cat food increased local ant density and toad mortality by more than 4-fold compared with unbaited sites. In some field trials, Ward Fear recorded meat ants killing up to roughly 90% of the baby cane toads emerging near the water's edge in baited zones. As Professor Shine explained to the press at the time, the method works by encouraging ants to flourish somewhere they already thrive, briefly advertising that there's abundant food nearby, so more ants show up exactly when the toads are most vulnerable. He also noted the toads' core weakness bluntly: their instinct to freeze and display their toxins, an ancestral defense against predators the toxin actually affects, does nothing against an ant that isn't affected by it at all. There's a second layer to why this timing matters so much. Cane toad metamorphs absorb water almost entirely through their skin rather than drinking it, which ties them tightly to the margins of ponds during the vulnerable window right after transformation from tadpole to toadlet. If ants can disrupt that narrow window, even just delaying or displacing toadlets from the water's edge for a few hours, heat and dehydration alone can finish what the ant started. Separate research on cane toad habitat use, including studies of how toads use hoofprints left by cattle around waterholes as temporary shelter, found that toads sheltering in steep-sided hoofprints were both more likely to be found by predatory ants and more likely to drown after heavy rain. Another example of how finely balanced the toad survival is at this life stage. There is an important limit to this method, and researchers have never claimed otherwise. Meat ants can only take down cane toads at the metamorph stage, tiny, freshly transformed toadlets a matter of days old. A fully grown adult cane toad can weigh close to 2 kilograms, has thick, leathery skin, and carries far larger toxin reserves. An ant colony poses no threat to an adult toad at all. What the ant strategy does instead is attack the population at its most fragile point in the life cycle, cutting down the number of toadlets that survive long enough to mature and reproduce. Given that a single female can lay tens of thousands of eggs per clutch, even a modest reduction in survival at the metamorph stage can meaningfully slow how fast a local population grows. Because the method requires nothing more exotic than supermarket cat food and existing native ant colonies, it has been picked up well beyond the original research team. Land management groups and conservation rangers in the Northern Territory have used bait-and-ant approaches to protect specific high-value sites, wetlands near vulnerable northern quoll populations, for instance, where losing ground to the toad invasion would carry a heavier ecological cost. The most remarkable part of this story isn't just that meat ants kill baby cane toads. It's what has been happening more broadly as different Australian species slowly work out how to deal with an animal that, on paper, should be nearly impossible to eat safely. The cane toads themselves are still evolving rapidly, and not just in leg length. Subsequent research, including CT scan-based studies of toad skeletons from populations at different points along the invasion front, has found that toads at the leading edge have developed narrower pelvises, longer forearms, and more compact, higher set skulls compared with toads from long-established populations near the original 1935 release sites. These aren't superficial differences. Breeding experiments have shown many of these traits are heritable, meaning the invasion front is, generation by generation, producing a version of the species increasingly built for speed and dispersal rather than for anything else. Researchers studying this process describe it as one of the best documented real-world examples of rapid evolutionary change in a vertebrate. Other native Australian species have separately been documented developing their own ways of dealing with cane toads. Some Australian crow and raptor species have been observed flipping toads onto their backs to feed from the underside, avoiding the toxin-secreting glands on the back and shoulders. Certain native rodent species in the Northern Territory, including the dusky rat and water rat studied in the Kimberley region, have been recorded eating cane toads while selectively avoiding or removing the most toxic organs. A level of behavioral precision researchers didn't expect from species with no prior exposure to this kind of toxic prey. Keelback snakes, a native colubrid, have shown some natural tolerance to cane toad toxins, likely inherited from ancestral lineages that were already exposed to toxic Asian toad species long before cane toads reached Australia, though even keelbacks generally prefer native frogs when they're available. None of these behaviors were common when cane toads first swept through a given region. They represent a slow, ongoing process of native wildlife encountering an unfamiliar toxic invader and, over successive years or generations, finding workarounds. The meat ant is simply the most effective and best studied example so far, a predator that didn't need to evolve tolerance at all, because its biology was never vulnerable to the toxin in the first place. It just needed the opportunity and a can of cat food to be in the right place at the right time. It's worth being honest about what this discovery is and what it isn't. Meat ants are not going to eliminate cane toads from Australia. Adult toads remain almost entirely outside the reach of any native predator, and the species population, spread, and reproductive output remain enormous. Ecologists studying biocontrol are also careful about a second-order question. Deliberately boosting meat ant activity around waterholes, even locally and temporarily, is not something to do indiscriminately. since meat ants are generalist predators that also interact with native insects, ground nesting birds, and other wildlife. Most of the research so far has focused the technique on defined, high-value conservation sites rather than broad landscape-scale deployment, and further work on any wider ecological trade-offs is ongoing. What the discovery does represent is something Australian conservation efforts had struggled to find for nearly a century: a low-cost, native, targeted method that measurably reduces cane toad survival at a critical life stage, without introducing a new species or relying on expensive infrastructure. It's a reminder that after decades of chasing large, engineered solutions, fences, traps, and gene editing proposals, one of the more effective tools turned out to be a predator that had been living alongside the problem the entire time, doing part of the job on its own, simply waiting for someone to notice and give it a better chance to work. The cane toad invasion is still one of the most consequential ecological disasters in Australia's modern history, and it isn't close to over. But for the first time in a long time, the balance at the water's edge has tilted, even slightly, in the other direction. And it happened because a handful of researchers watched what an ordinary ant colony was already doing, and asked how to help it do more of it.